So, as you probably know, human genes, the genes are actually quite large, but the coding regions that actually encode the proteins, shown in red here, are called exons, and they're very short, and they get put together by this complicated process of RNA splicing. So, over here we have a blue gene with 3 of these blue segments that are going to get put together. And these long black regions called introns, they're not going to end up coding for a protein. So it's long been thought that if you inserted something in there, that's all going to be removed anyway and thrown away. So if you insert something in there, it's not going to have any effect. But increasingly, what we're finding is that when you insert a large piece of DNA in the middle of one of these introns, it can have all kinds of unexpected effects on how much of the messenger RNA for that gene is actually made, how much of the protein is made in the right structure, and so on. And further, even if the insertion is outside of the gene, there are some instances in which you can have an inhibitory effect at a distance of an insertion like that. So what we're seeking to find is instances of these two over here, and trying to find out how common that is and how commonly might they have an impact on human phenotypes. Okay, so Dave has already alluded to a very hot topic in human genetics today, which are so-called complex traits. So this is in contradistinction to the Mendelian traits, which are caused by a single mutation in a single gene. So for example, height. Okay? We see a lot of genetic variation in height. We know that height runs in families, but it's not governed by a single gene. It's governed by interactions between many genes. And very importantly, from the perspective of medicine, susceptibility to most of the major killers of humans is a similar sort of complex trait. It's not governed by a single gene, but it's governed by many genes, and small changes in the behavior of those genes, when put together in the right combination, underlie these susceptibilities. And so I've just listed a couple of cases: cancer susceptibility, neuropsychiatric, cardiovascular, and you're going to hear about a lot more later today. So again, our hypothesis here is that these insertions can knock down the gene expression somewhat randomly in some subset of genes, and perhaps this is going to be a significant piece of human mutation that's been overlooked until now. Okay, so David has already alluded to this, so I'm not going to spend any time on it, but you can go out and get your genome sequenced right now. A lot of us feel like as we move to electronic patient records that we will see human genome information attached to that electronic patient record. Probably it'll be routine within this decade. Okay, so I want to finish by talking about what I see as really the next frontier in genomics, which is going from reading the DNA or sequencing it to synthesizing the DNA or writing it. So this technology is currently in a growth phase as well in terms of what we can do per unit cost. And there's this new field called synthetic biology. Okay, you'll remember synthetic chemistry and you'll remember biology, but here this is kind of a marriage between engineering and biology. And as I'll show you in a moment, undergraduates as well as others worldwide are organizing into teams to make so-called biological devices. I'll show you an example of that. Greg Venter comes up again because he's really one of the leaders in this change and has synthesized, as you know, entire bacterial genomes, and he's hoping to harness that technology to make biofuels and other high-value chemicals. At Hopkins, we are actually working on synthesizing a modified version of the yeast Saccharomyces cerevisiae, the one that you use to make bread and beer. A modified version of that using an undergraduate workforce. This is what we call the Build a Genome course at Johns Hopkins. So the IGEM, or International Genetically Engineered Machines competition, is held at MIT every year, and it's been going for about 10 years now. And as I said, this is an annual competition, and teams form at different universities. Generally, the teams consist of undergrads mostly interested in biology or engineering, but increasingly also people interested in the human practices side of this field of synthetic biology. So how does the public perceive this unholy marriage of synthetic and biology? What are the biosafety and biosecurity concerns? What are the possible benefits, public health, etc.? And what you see here is actually a plate which is seeded with a layer of bacteria. And the bacteria have essentially taken a photograph. They're engineered so that when you shine light on part of the bacteria, they change color. And so, hello world is a pun. You may know that that was the first thing that a computer ever said. So this was a synthetic biology pun. This was done by a team of undergrads at the first IGEM competition. This is another fun thing done by an IGEM team in which they created shapes by strategically positioning bacteria that are engineered to fluoresce different colors depending on their chemical environment. So these three different spots here have some chemical in them that's diffusing out into the agar, and when the concentration is high, you get this red color, and then when it gets higher, you get no color, and then when it gets higher again, you get green color. So anyway, these are examples of these IGEM competition projects that involve putting together small circuits built entirely from synthetic DNA. Each of these circuits involves anywhere from 5 to maybe 50 genes, so they're relatively simple. And I can tell you, they're also it's actually very difficult to do this well. So what are we doing? Well, even though I'm working on human transposons, I'm trained as a yeast geneticist, and I work on Saccharomyces cerevisiae. This organism is my favorite. Ben Franklin loved it too. This is, quote, "Beer is living proof that God loves us and wants us to be happy." This is actually the first slide I use for the undergrads in my course, telling them how yeast and humans have been intimately related for a very long time. This is a 1 kilogram chunk of yeast that you can buy, and it's approximately 1,000 times the world's human population. So there's a tremendous power in these very large numbers.